It's my pleasure to contribute. Again, thank you for inviting me. I will be talking about trial readiness and what do we need before a successful clinical trial in MPAN. Let me start by saying drug development is complicated. And there are various challenges and obstacles along the way. And during my talk in the next 20 to 25 minutes or so, I want to discuss a few of these, and that includes we need to understand the pathophysiology, we need to find a good drug candidate and identify and validate biomarkers. We need to find good clinical outcome measures. We need to improve our knowledge of the disease and the disease course. We need effective recruitment strategies. We need to choose an optimal trial design. We have to face various regulatory aspects, and we need funding to do all this. So let's have a closer look. What's our challenge? Challenge number one, we need to understand disease pathophysiology. Thankfully, there are a lot of things we already know, and we've heard about this today, but there are still a lot of things that we don't know. And this includes how do mutations lead to neurodegeneration? Why does iron accumulate? What is the role of iron? And there are many other questions where we still don't know the answers. So I think one of the things we need to do in the future is to learn more about this. And I know that there's a lot of effort being made, and there are a lot of efficient people here today and out there, and that's good. Challenge number two, we need a good drug candidate. There's a formal drug discovery process, and you can see an icon on the right, and that includes target validation, assay development, high throughput screening, and this may also identify a drug which is already on the market in the sense of repurposing, where we can find a hit to lead, so an optimal drug candidate which may be most potential to have benefit, which will then be optimized. We have to do various preclinical studies and clinical studies in this process, and that not only includes the biochemical aspects as such, but also various regulatory things when it's about facing standard manufacturing procedures for the formulation and looking at the guidelines on process validation for finishing products when we apply for approval to regulatory bodies. You are all familiar with the different clinical stages of drug studies, and that includes phase 1 to 4, where we look at safety and dosing, first in healthy volunteers. We then study our drug in a small number of patients, followed by a larger number of patients with the targeted disease. We need to identify and validate biomarkers, and biomarkers are objective indicators of medical states which can be observed from outside the patient. Biomarkers should reflect target engagement and response to treatment. They must be accurate, sensitive, reliable, and reproducible. They may be biospecimen based, so they may be markers in blood, urine, CSF, or biopsy material. And they may be molecular, genomic, or cellular. We can also look at clinical or imaging markers. Biobanks are the ideal hub to collect specimen and longitudinal data. And again, the initiatives that we see in the NBIA fields are very good with regards to these biobanks. The fourth challenge is that we need good clinical outcome measures, which are measurable changes in health, function, or quality in life that result from our treatment intervention. And we could consider using functional outcome measures, for example, how good a patient can do a walking test. We can also do imaging markers, for example, the iron accumulation in the brain, or look at changes of blood markers or gene expression. We do have to be aware that some of these outcome measures may not be specific enough to determine treatment effects, so we have to validate these. In recent years, patient-centered and patient-reported outcome measures have drawn a lot of attention because they are grounded in what matters most to patients. In fact, there is also a patient-centered outcomes research institute, and they have a rare disease advisory panel. There are different categories and examples of patient-reported outcome measures listed here. So the categories include assessment of disease burden and level of unmet needs. You could assess treatment impact, patient perception of change, or the meaningfulness of perceived change. And there are various examples of these patient-reported outcome measures on the right. You can also use different instruments to measure these patient-reported outcome measures. So they may be rare disease specific, in that they have been developed for a particular rare disease and then used in a trial for that disease. An example would be the scale of assessment, the scale to assess the activities of daily living in PCAN, or the clinical rating scale in PCAN, which you will all be familiar with. You can also use a non-rare disease specific scale, which has been developed for a disease that is not the rare disease you're looking at, but is then used in your rare disease clinical trial. For example, the UPDRS, which was used in the deferiprone trial for PCAN. And then there are also generic or study specific rating scales. So I think what we need to do in the future is to identify and characterize our biomarkers and outcome measures which may be suitable for an MPAN trial. And we need to develop the MPAN specific rating scales, and it's exciting that Marta is doing that at the moment, so I look forward for this to be published once she has validated it so it can be used in a broader group of patients. The fifth challenge is that we need solid knowledge of the disease. This includes that we can estimate the magnitude of the problem, determine the underlying incidence and prevalence rate, and this has an asterisk because this is important when applying to the regulatory bodies later on. We need knowledge of the range of disease manifestations. So what's the clinical variability? Again, we've heard about that in earlier talks, that there is in fact a broad clinical variability in MPAN, but also in the other NBIA disorders. We need knowledge on genotype-phenotype correlations. Again, I was excited to see this during the meeting. We heard about this in PCAN on the first day. We also heard about this today by Susan Hayflick when she showed her genotype correlations. We also need solid data about disease progression over time, so natural history, and I spoke about that earlier. And we need to identify the stages of the disease progression that may be responsive to treatment. So should our future treatment be given in the mid stage of the disease? Will it also be helpful in late stages of the disease, or will it then be too late for treatment? Or is it something that we could give in early stages or even pre-manifest stages to prevent the onset? I've already talked about the importance of natural history, and that is because this will allow us to use certain clinical trial designs, and because the regulatory bodies request solid data. I've shown you this guideline already by the FDA as to how to perform a solid natural history study. And I mentioned the current paper, the most recent paper from this Chinese group about the natural history in PCAN and our own data as well. So again, I think we need to perform a solid natural history study in MPAN. We also need effective recruitment strategies, and that includes various aspects. That includes that we need to be able to identify our patients. This can be done by enhancing clinical expertise so that clinicians will recognize the condition early. They also need access to provide genetic testing. And it's interesting that in other genetic diseases, there's new initiatives, and I want to share this with you, the PD Generation Initiative, which is being offered to patients with Parkinson's disease who can now be tested free of costs and receive free counseling in various centers across the United States as part of the PD Generation Initiative. It's also interesting to see that there are now strategic partnerships between pharmaceutical industry and diagnostic companies in order to facilitate recruitment for future clinical trials, given that future clinical trials will be based or will allow patient inclusion based on their molecular phenotype. Effective recruitment strategies also includes that we raise awareness and that we treasure the value of our rare disease communities. The rare disease communities, which consist of patients and their families, the advocacy groups, the networks, the registries, and the small number of therapeutic area experts. So basically all the people that are here today, and I think the NBIA community is really one of the best examples as to how the rare disease community can be very efficient. Raising awareness can never be done enough, and other rare disease groups have been publishing about this. For example, Lewington and colleagues discuss the why and how to raise awareness and who should be the target audience. Effective recruitment strategies furthermore includes that we build solid clinical research infrastructures with clinical centers of expertise, where we train a study team and create resources to support that trial readiness. And then finally, effective recruitment strategy also includes patient participation itself. And it's interesting to see what drives patients or what motivates patients to participate in clinical studies. And here's a paper about their decision-making when it comes to contributing to a biobank, and how important the patient-recruiter relationship is in this regard. There's another paper from the Parkinson's field I would like to share with you, where the authors asked patients and their caregivers to name the 5 most important reasons why they may not take part in a clinical trial, or to list the barriers to an effective clinical trial. And you can see the answers on the left. And this includes that patients are worried about potential side effects, about the disruption to their current medication, that they may be given a placebo, that there is upheaval to their life when taking part in a trial, that they wish to be kept fully informed, and that they're worried that participating in one trial may exclude them from taking part in another. So I think it is important that we bring our patients and their families to the table early on in the development of the life cycle of drug development, so that we consider these issues and address these. The next challenge is that we need to choose a suitable trial design. And this is because standard trial designs are not ideal to obtain adequate safety and efficacy data when the cohort study is small. So we have to consider alternative designs without compromising the validity or power of the trial. There are various research models available now that can be utilized in small cohorts, so for rare disease research, and you can find a list here. And the selection of the design eventually depends on the choice of controls that you can use, the choice of the outcome, and the nature of the disorder. But clearly this choice should be driven by the defined study question and nothing else. There are various options with regard to the controls. You can either go with no controls, and that applies to diseases where you have a devastating prognosis in the absence of treatment. Or you can use historic controls, where you have a small or rare disease population where the natural history and the outcome without treatment is well known. And I think this is the option that applies to the NBIA field. We have a rare disease, but we need solid natural history data information. And that is why, again, I've talked about this again and again, why we need solid natural history data. Eventually, you can also use longitudinal outcome measures, which include repeated observation of a few variables, and ultimately this requires the smallest sample size. If you want to learn more about the recommendations for designs of small population clinical trials, you'll find information online, including this paper here. With regards to the analysis of data, this is also very complex, so you need an expert team of statisticians. There is the increasing that people share their data and stop keeping their data silos. And we need efficient data sharing ecosystems for this, or making data public, of course, protecting the health information of the individual patient. This is also because multiple analysis and reanalysis of data in the future may lead to new results and new conclusions. The eighth challenge is that there are various regulatory aspects on the way, because the regulatory bodies have high standards for the sponsor to demonstrate effectiveness and safety. You're all familiar with the regulatory bodies, which include the FDA in America and the EMA, the European Medicines Agency in Europe, there's also a global architecture called the ICMRA, the International Coalition of Medicines Regulatory Authorities, which provides this global architecture to support enhanced communication and information sharing between their members. The EMA itself also has a sub-team for the study of orphan drugs, which reviews the applications that seek orphan drug designation. The formal designation criteria of orphan drugs are listed in the regulations mentioned on the lower right, so in the EMA and the FDA regulation. And basically, this applies to conditions which are rare, so they affect less than 5 and 10,000 people based on the prevalence criterion, and the fact that there exists no satisfactory treatment, and that the new drug, so the drug that is being studied, may produce significant benefit over those which currently exist. To facilitate the development of new drugs, these regulatory bodies have various incentives, and that includes protocol assistance to maximize the chance of success, where you have a closer communication with these regulatory bodies. They allow access to the centralized authorization procedure, so that you don't need to apply in multiple times from all the various member states, but you can apply to only one regulatory bodies, which result in a single opinion and a single decision, which will then be valid in all EU member states. The orphan incentives also include market exclusivity and regulatory flexibility, which I'll come to in a second, as well as fee reductions. The regulatory flexibility has these different points. So for example, there's fast track designation, which is meant to accelerate the review of drugs which can treat serious conditions. And serious is defined by the fact that the condition has an impact on survival, day-to-day functioning, or the likelihood that the condition will progress if left untreated. And the examples given on the FDA website are HIV-associated AIDS, Alzheimer's disease, or epilepsy. So I would believe that NBIA would also fall into this category. Another flexibility measure is the breakthrough therapy, which applies to drugs which may demonstrate substantial improvement over available therapies. And substantial is defined by the magnitude of the treatment effect, in that a drug should show a clear advantage over the available therapy. And the other instruments are accelerated approval and priority review. The ninth challenge is that we need funding for all this. Not only does drug development take very long, so it takes 13 years for a drug to develop for neurodegenerative disorders, but it is also very expensive. And it has been estimated that the development of a drug for neurodegenerative disease costs more than $5 billion US. I am, in fact, intrigued by the fact that this is higher by the factor 7 compared to the development of a cancer drug, and I cannot answer as to why that may be the case. However, I do understand that sponsors therefore have to perform a economic evaluation before they move into the developmental field, where they consider cost and effectiveness in that how much does it cost them to produce this drug in comparison to how effective it will be for them. This is why hundreds of thousands of potential drugs die on the way. And there is the term called the valley of death, which is the gap between the academic and the clinical research side, where the drugs die in the valley of death because of lack of funding. We are all familiar with the traditional sources of funding, including the Horizon Europe program by the EU, as well as the grants by the FDA and the other international instruments. We are also very grateful for other sources of funding, including patient organizations and private donors. But it is interesting to see that there are also new funding instruments which the medical and basic scientists may not be aware of, and that includes mega funds and crowdfunding. And this is there are also industries who publish this as to where you can find where you can find this new sort of funding. The idea is that you de-risk the drug discovery process for the small and medium-sized enterprises, so that even startup companies can go into this process. So I want to say, let's get ready for a successful MPAN clinical trial. We can find recommendations for small population clinical trials online, not only by the academian side, but also by the industry side. And the paper on the lower left was published by McKinsey and Company, who also look into this because this is really important for society. And we can also learn from the failures from previous studies, and there are reasons as to why these trials failed, and we should consider this when planning our future clinical trial. So the future steps, I believe, that we should keep in focus is that we do a landscape assessment as to what we need in the future and where we should put our priorities for future research. We can then develop a program and then execute a program so that our runner will overcome all the obstacles on the way and then reach the finish line, and that we will have a drug available for our patients in the very near future. Thank you.